All right.
And it's all yours. Okay.
All right. Thank you. So let me come back over here and all right. So here you remember that we had some instructions for season 8, which was the first part of the single cell analysis. And if you click on that, we had the overview over here. So we started with single cell analysis. We introduced the sequencing technique, how we can process that in Dolphin Next, and how those result files of the Dolphin Next pipeline and the Cell Ranger pipeline actually looks like, and how we can use that to import UMI tables to our sessions and start data analyzing of the single cell data. So we have been using primarily the Surat package. This code, if you don't have the package itself, it's going to install, but if you have it, it's going to load it to your R environment directly. That is, if it's asking if you should, download from the source, you can say no, and if it's asking for you to update old packages, that is Surat depending on, you can just say A as in all and install all of them. The example that we've been using for single cell analysis was this small PBMC data with somewhat around 1,000 cells. And we're going to use that for analysis. Today, we're going to focus on identifying those cell types within this PBMC data and try to figure out if we can find T cells, natural killer cells, or other cells like monocytes and different types of cell types that you can find in PBMCs. So that data set is actually here, the folder that we ask for you to copy to your project space. So you can find the actual folder here as well. And what we actually want you to do is to get this folder and transfer to your local computer. You can use it. You can do that by FileZilla. You can just choose the folder and toss it in your desktop really quickly. And there are some instructions to how to connect to FileZilla. Then what you have to do, I would like you to open RStudio, and we'll be here for the remainder of this lecture. So the first thing that I want you to do when using R, I want you to set your working directory. This is really important because when we ask for certain files to be loaded in our environment, we have to be in the right place. So in this case, I want this to be really simple, and I want to choose the desktop as the directory. So the only thing that you have to do, you have to go to the session, set working directory, and choose directory. Now, the only thing that you have to do is to go to your desktop and say open. As you can see, the moment that we did that, we actually ran a code that says set WD desktop, meaning that set working directory desktop. So every file that we're going to call directly will be searched in the desktop, and that is how we are going to start coding. So the next thing that I want you to do, I want you to create a new file. So in order to do that, you have to click over here on the top left, new file. You have to click on R script, right? Then you're gonna, come up with this open new script. So this is untitled now. You have to save it in order to start. So the only thing you have to do, you have to save. So what I want you to do, I want you to save it as main 1K something. And you don't have to say .r. It's going to save it as a R file anyway. So it already exists, so I'm not going to click save, but I'm just going to find the one that I've used. Okay. So as you can see, the folder is over here. We already copied that to our desktop, and we set our working directory as the desktop. So if you type in the path like this, it's going to understand that you are searching for this folder in the desktop, and you'll immediately be able to import the UMI table as we've been discussing. So I'm just going to copy these really quickly to both Slack and Zoom possibly, so that you guys actually see what I'm doing. By the way, I have Slack on my right and you on the left. So whenever you guys want to ask a question, I'll be able to see it. So don't mind stopping me if I'm going fast or if anything is not clear. So I'm just going to copy paste this entire code. And what you can do is that you can actually use this to copy to your file, our script that you just created, and we're going to work from there. So for those of you who couldn't transfer the folder from your cluster space, you can use this alternative method, and it's going to just import the same UMI table as we have done over here. So we have some alternatives if you couldn't do that. So let's start and try to understand what we are doing over here. So the first thing I'm going to do, I'm going to load the library. I'm going to first cover the last section that we have done on Tuesday. Then I'm going to quickly talk about the homework, and we will move on with the next session. Okay. So is anyone not clear about this? Are we good? Okay. Let me get this larger. Yeah. Okay, so the next thing that we have to do, let's see what we have imported. You can either use the import function, read 10x, which actually imports the UMI table as a result of the cell ranger pipeline, and puts that as a matrix format, right? So this is a quick way of looking at the matrix itself. So what we have over here is that we have a table of some 36,000 features or genes, and some number of columns that are the barcodes. So in this kind of a matrix, which will be usually the case for most UMI tables, regardless of the fact that you are using 10x or cell ranger or any other single cell pipeline, you're going to have usually the barcodes as the columns and the genes as rows. So we have done this with bulk as well. In bulk, when we covered that with Alper, the biological samples were usually on the column. So this is basically the same case, but we have barcodes instead of biological replicas, right? So as you can see, there are some dots over here. Dots meaning there are just zero. This is normal for single cell because most of the genes wouldn't have any expression in any particular cell. But I mean, there are thousands of numbers over here. Even though we don't see them, they actually have substantial amount of UMIs. So in order to take a look at the UMI distribution and the number of counts, we have different kind of methods, and that's what we are going to do today. So the first thing that you have to do after having this matrix, you have to turn that into a Seurat object, right? So let me quickly do that and try to see what we can do with it. So I'm going to appoint the Seurat version of the matrix to a different name in order to differentiate them. I don't want to overwrite it, but you can do that as well. But in this case, you will have to import the same data again. So it's up to you. You can just write the same thing. It really doesn't matter. But I mean, you may need to keep it in a different way. Most of the time, if there's a Seurat version of a UMI table, I'm actually writing something SEU in the end to indicate that this is actually a Seurat object of the matrix that we are working on. So I'm just going to click on that and going to see what we actually have done. Okay, so this is the this is the result when you just click on the object and just press enter. It's printing some summary of what is inside the Surat object. We don't really see much now because we haven't really done anything. So today what we're going to do is that we're going to keep populating different kind of analysis in the same object, and we are going to put everything in the same object so that we don't actually use any other tool as well, tool either. And what's going to happen is that everything will be in the same object. So that's why I'm calling it an object, because it's not a matrix. It's not a any kind of vector or something like that. It's a collection of different kind of matrices and vectors, so that you can easily do the analysis with only using one object. And this is one of the advantages of R when you're doing some kind of an analysis. Usually people wrapping up built-in functions for you to do complex analysis like single cell. They come up with packages and they create objects like that in order to make people easily analyze their data without even thinking of creating different objects and other stuff. Of course, we're going to create some other objects, but eventually we'll be using this Surat object only. So let's take a look at this object a little bit more in detail. So one of the things that you can do now is that you can view the metadata. Okay, we have a count matrix, but we will have a metadata, an additional data table that gives more information on each barcode. So in order to do that, just write view with V being capital letters, and just click on PBMC1K Surat and just click add. When you click add, you have multiple options to choose from. Here you have to choose metadata in order to view that table. So if you click on that, an additional panel over here will open, and it will tell you some additional options about each barcode. So basically, the first thing that we have when we create a Surat object here with this command, what's happening is that the Surat package calculates the total UMI of all cells and the total number of genes or features that are non-zero by default. So basically, you can use these measures directly as a quality control process, and we'll be able to take a look at each cell and their total number of UMIs in order to understand what we can do and which of those cells we can remove. So let's get on with it, and let's try to understand what's actually in them. So one of the first functions that you can use is the violin plot, right? So what violin plot is asking is the object itself, and this will be the case for most of the functions that you use in Surat. The first parameter of all functions, there's a question. Oh, yes. Okay, let me remind you that again. So ncount RNA is the total UMI count of each cell, but nfuture is the total number of non-zero genes in a cell. So how many?
Thank you.
Make sense?
Yes, thanks.
Okay. Because it's really important, because sometimes you may have some high number of UMIs from really low number of genes, and that may actually indicate that even though the UMI count is really high, that cell may not be really informative. So it's better to keep both. Okay. So the first thing that we're going to do, we're just going to run violin plot. So you'll get some kind of an overview of the distribution of the UMIs summation and the number of genes here. So what I'm seeing actually is that on average, each cell has somewhere around maybe 6,000–7,000 UMIs, total UMIs, but there are some cells that have extremely high number of UMIs. So there are multiple indications of those. So what could be happening is that you have some doublets. We talked about single cell RNA-seq and the droplet methods, and it is highly likely that even though you have a highly optimized microfluidic system, you may actually accidentally encapsulate two cells in a single droplet. So that may actually indicate that you have actually two phenotypes, maybe a T cell and a NK cell within the same within the same droplet, and that may actually hinder your analysis. So we actually don't want them, and we're gonna cut them off. To better view, you can use the second code over here, the feature scatter. So it basically creates a scatter plot with two features that you choose from the metadata. So as you may remember, this two encount RNA and feature RNA, they were both in the metadata. And the feature over here actually refers to the features in the metadata. And you can call some genes with that as well, but you have to be careful. So we're just going to click on this and run. So not only now, I see the distribution of UMI summation, but I also see what is the equivalent number of genes in them. So basically, not only that the cells have a lot of UMIs, they actually representing a lot of genes as well. So I mean, this might be for you, a better indication that these cells are actually doublets, meaning a droplet that has more than one cell in it. So this was really clear. So before moving on to filtering out these cells, we are going to introduce additional material. So as we've been discussing on the last session, that it is highly likely if the cells are in apoptotic state, they may express, we might have been capturing some RNA from the mitochondria. So what's happening is that you'll be seeing a lot of reads aligning to genes starting with MT dash. And what we are really actually doing over here, we are calculating the total number of expression and UMIs from genes MT dash divided by the total cell for each cell, divided by the total UMI count for each cell. So if we just click on this, what's doing over here is that's calculating the percentage of MT expression for each of those cells, and it's putting it as a different column in the metadata. So basically what's happening over here, it's actually representing why Seurat is so efficient. So normally in a data frame or a data table, this would indicate a column, but we don't just have a data table in Seurat. We have multiple tables. We have count matrices and we have metadata. What's happening is that if you call a new name of a column of the Seurat object, it automatically appoints that to the metadata, but not to the count metrics. So this is one of the advantages. There are a lot of things that are happening automatic, that the only thing that you have to worry about is the analysis itself. So we are going to do that. I'm going to just run this really quickly and see what has changed in the metadata. Sometimes checking if I'm getting any questions. So as you can see now, originally we have the total UV count and the Gini, the number of non-zero genes, but now we have the empty percentages as well. So basically what we are trying to understand, if there are some cells, even though they have substantial amount of UMIs and some number of genes, their mitochondrial RNA percentage is actually extremely high, which may be indicating that some of those cells are in apoptotic state. So after doing that, I'm going to add a new input to the future parameter here to the violin plot. I'm going to just run the same violin plot function. I'm just adding the percent empty as well. So whatever the number of parameters, the features that you add over here, they are all going to be visualized right next to each other. So you can just keep populating this names over here. So I'm just going to run this really quickly. So this two, total UMI violin plot and Gini violin plot were the same, but we just added the mitochondrial percentage as well. So basically, as you can see, that some of these cells are actually having only empty Gini expression, which may indicate that they have nothing else to present other than empty phenotype over here. So question. Just by looking at the violin plot, and I really want somebody to answer this, by the way. So what do you think should be the cut over here based on the MT?
Maybe 20%.
No, great. And how do you understand that, Alicia?
But just looking at the dots, the majority of them are situated in the thicker part of the plot. So it's around, so it's under the 25, but for maybe making it even a little more stringent and putting 20, but perhaps 25 would be a good starting point. I don't know.
Yeah, okay. Yeah, well, I mean, here's the thing. So in single cell analysis, there is no best answer. It is always possible that, well, I mean, just by removing those that are higher than 20, you may be losing some cells that are of some importance. Maybe we can try Olisia's option as well. And Kashi also asked me something, I believe, if we have to remove them. Yes, you don't necessarily have to remove them all the time. So it's based on what you're expecting from your sample. So some samples from different cases, like I don't quite remember right now, maybe in cancer, that mtRNA expression may indicate some other phenotype that you need to be capturing, but it's based on the analysis. In this case, you don't really have to remove, even though for a variety of cases, MT percentage is expected, is considered to be a regular quality control measure that you have to be mindful of. There's also the possibility that if you keep the cells that have high MTs, they may actually form clusters itself because they are actually expressing some kind of a phenotype. And that might be the case. Maybe you don't really have to remove them. So there's actually no best answer here. It all depends on, if you can come up with your clusters, if you can figure out your cell types. And that's what we basically want to do. There are different kind of methods. Okay, but great, Olsia. Thank you for answering that beautifully.
Sure, I actually have a quick question. What's the difference between the M-count RNA and M-feature RNA? I think you mentioned, but I missed it.
This one? This too?
Yeah.
Okay, so N-count RNA is the total UMI of each cell. But N-feature RNA is the number of non-zero genes in that cell. So maybe in a gene, you may have 10 UMIs. In another cell, in another gene, you may have 20 UMIs, right? So what's actually discounting is the non-zero counts in each cell. But this is the total UMI count.
Okay, thank you.
Mm-hmm. All right. So but by using these terms, we actually figured out that maybe we have to delete cells that has less than 200 genes, and we check that by looking at the metadata. So there are a lot of things that you can do with this kind of a view of tables. So we have done this by viewing the metadata here. Just going to quickly do that. So what you can do now, you actually can sort cells based on each of these columns. Just click on, for example, M-feature RNA. So here, when you click it, you will see that, in a sorted way from bottom to top, and you'll see that there are some cells that actually have, just a handful of, genes that are non-zero, right? And their total UMI count is 500. This number could be good, and it actually passed Cell Ranger Pipeline's quality control measures. But what's really important is that if you trust a cell that only whose 13 of genes are actually non-zero. So if you could go down a little bit, you'll see that the majority of cells actually have a quite high number of non-zero genes in them, right? And this may actually indicate that some of these cells may not be really that good. So it's again up to you. We will see how we are clustering single cell data sets, but this may be indicating that these cells may not really be important. So in this case, we are going to be removing all the cells whose N-feature RNA, the gene count, the non-zero gene count, lower than 200 by selecting those that are higher than 200. Basically, we are just taking a subset. Is there any right threshold for N count and N feature? Very hard to find out whether these numbers are real or are artifact. It's a really good question. So it's a trial and error process. basically, most of the time when you have really low UME threshold, you may actually start seeing some clusters that do not necessarily have any unique markers that are specific to them, indicating their phenotype or indicating their cell type. That may actually give you a hint that you may actually increase your UME threshold. So again, these numbers are never absolute. You have to remove, add more, you have to go back, select another threshold until you believe that you've been capturing the right amount of cells. But yes, you're right. So it's really hard to tell that. It's for all biological problems, it could be different. So we are just going on with these thresholds just for an example. And also what we also talked about was that some of the cells could be multiplets. So I'm going to remove all the cells that have more than 20,000 UME in total as well. So yes, so after introducing all these, filtering controls, we are going to remove these cells. So as you can see, now we have some 1,200 number of cells now, and I'm just going to take a subset and overwrite the object. Now let's take a look at it again. What you're seeing over here is that now we have around 1,000. So basically somewhere around 100 cells couldn't pass these thresholds, and so we removed them, and we're going to keep moving forward with these cells alone. So this was where we left off last week. Did anybody try doing the homework too in the last session? So if you go to the 8th session, at the end of the 8th session, we had another example, which was the 3,000 cell version of the data that we've been using. So you had to basically do the same thing on it as well. I have an example of that too. Let me quickly open it. So basically the workflow is quite the same, right? So the first things that we do, we kind of take a look at the violin plot, maybe scatter plot, calculate the empty percentage, and come up with some thresholds. In this case, I've chosen 5% as the percent empty threshold, but again, it's up to you, as we've been talking with Alicia. Now, sometimes I've been heavily criticized for using 5, but what's happening to me in that case was that if I keep it to 20, I definitely see clusters that only have empty RNA expression in it, but it may not have been the case. So for your sample, for different applications and experiments, this threshold will be changing. So you have to be mindful of that. So it requires some kind of biological interpretation. Okay. So I'll be moving forward with slightly with normalization, and we'll moving forward with the next session's content. So the next thing, after filtering out those cells, we have to make sure that we are accounting for sequencing depth. So for doing that, we're going to normalize. So basically what this function is doing is that it's incorporating several different normalization methods for your raw RNA count, and it normalizes that. So I like to first make you take a look at the part where we are storing the raw RNA counts in the Surat object. So if you go with your object, you have to click on @ and you have to choose assays. After you choose assays, you have to click on $, which is going to make you choose which assay that you want to work on. So Surat is a highly functional package. It allows you to analyze multiple assays in the same time. In this case, we only have RNA, so we only have the RNA option. Now we're going to click on @ again. So as you can see, the Surat object has a lot of levels. And you have to just click on counts to see the row counts that we have imported in the first place. So it's the same metrics as we worked on, as you can see in the beginning, right? So it's the same as this object. So basically we are taking the row counts anywhere we go. So one question that you guys may have, what will happen if I were to normalize my data? Okay, will I lose the original counts? Well, the beauty with Surat is that you actually don't really have to do that. The normalized version and the raw version are actually stored in different parts of the object. So now I'm going to just click on something else. I'm just going to remove counts and see there are multiple options over here. If you click on the data, this was supposed to be the normalized data, but we haven't normalized it yet, right? So the counts and the data is the same, but once we run the normalized data function, this data will be replaced by the normalized version, but the counts component will be changed. So let's go with that. Let's click on it and run it. As you can see, every time I run a function, I appoint the result of the function as the object itself. So I'm overriding at each step, I'm actually overriding to the same object. And I'm filling another component in the object, and I'm still using the same object. So this is the way we need to be coding in Surat. So when I do that, it quickly normalizes. This data set is quite small. So this loading, printing screen actually happened really fast. But for some data sets where you may have over 10,000 or 100,000 cells, you may actually wait for each of these steps. So what happened? Let's take a look at the same part. Again, you type the Surat object, you click on essays, then you click on RNA, then you click at again, but now you're going to click on data. And as you see that we don't have row counts now, we have some normalized counts. But again, if you take a look at the counts, instead of the data, we still keep the row counts here. So it's quite interesting. So you can basically, the fun with the Surat is that you can keep running the same code and you're not normalizing the data that you have already normalized, just taking the data from counts and pointing to the data, but nothing really happens. You're just doing the same thing. Nothing is actually changing. This is really nice, actually. If you may confuse the order of the functions, sometimes you need to start from the beginning, but in this case, for a lot of functions, you don't really have to do it, right? Okay, so this is where we left off, right? So one thing that I'm just going to add was that you can, for each of the functions in Surat, you can quickly type normalized data, help, and see different things that you can do with the function. There's a nice description for all the functions in R, and this is the appropriate way of coming up with packages. And Surat being one of the most popular single cell analysis packages, their descriptions and documentation are actually quite really good. So what you have to do, just go down and see different things that you can do with it. So basically, as you can see, we didn't actually introduce much parameters because most of the parameters actually have default values, right? So how we are normalizing in default with Surat is that we are dividing all the UMIs of each genes of each cell by the total number of UMIs of each cell. Then we multiplied that number with some 10,000 in order to normalize the sequencing depth, right? Then we take a log of it. So this will be coming up with a scale, we'll multiply the divided UMI counts with scale factor, then I'm going to take a log of it. But you can try actually different things as well. This normalization method, for example, is specific to a different assay other than RNA, but technically you can use on it on genes, but it's not preferable. So the only thing that you would do in this case, you would just go ahead and define an additional parameter. Basically, if you type several first letters of some parameter, it's going to pop up in your RStudio in pane. So you're just going to click on it and you can define the parameter yourself, like it's been shown in the. Some reason I cannot click. Yeah, okay. It's in the help function or help page over here, but we don't really need it now. The default version is okay. Again, I'm just going to click on it, nothing will happen. So let's move on with the next section now. Everything is clear until now, guys? Any problem, anything to ask? So if you go to the main page of the bootcamp, you can see a quick link to the second part of this single cell analysis, today's session. Okay. So we have covered these today. We've seen how to normalize and filter, but now we are going to start with what I believe is the more mathematical part, and what I believe that requires a lot of explanation before going forward, because we have to understand the essence of single cell analysis and why we are actually using clustering in the beginning. So as a statistician and machine learning scientist, I actually started doing biology like a few years ago. I believe that single cell analysis is one of the most beautiful applications that you can do clustering with. So Nazgul is asking, can we cut off the data and do filtering after normalization? So when you, interesting question. So when you do normalization, you can filter still using those quality control measures that we have used, and it's going to be using the total UMI count calculated based on the row count. So yes, you can, but I don't suggest doing that. The proper way of doing it would be to filter first and normalize later. But yes, you can.
Can I ask a question? So in the normalization step, I might not have understood it well, but what is the normalization to? Like, is it to housekeepers? How or is it like a normalization of the counts?
You're normalizing to the total UMI count of each cell. Basically, let's say that you have total, 200 UMIs in one cell, but in another cell it's 400. Some genes, even though they may look like you have 40 genes in one cell, but 20 UMI count in another gene of another cell, it may look like that gene actually has a higher expression, but in reality, the total UMI actually it's based on the total UMI. It has nothing to do with that having higher expression. So that's why we're normalizing the total UMI count before we move on.
All right, thank you.
Okay. Right. Any other question? All right. Okay, so why data clustering is essential for single cell analysis? So we talked about droplet methods, right? So we have a really interesting parallel single cell RNA barcoding system where we have droplets, where we capture the bead that has the barcodes and the cell in it, so that we know the origin of each RNA, that we know that, okay, there's a barcode over there. All these RNA are from that cell. So we have these amplified version of the UMI table where we don't have just bulk data, but we have all the cells as well. So basically what's happening over here is that you don't really know the origin of each cell, right? You know that it's a cell, but you don't know if, maybe CD3s are higher in that cell, which may indicate that those could be T cells. For those of you who know skin biology, maybe those cells are of keratinocytes, so their keratin genes will be much higher, and we can't really immediately see it because this table over here is actually quite large. You maybe, take a look at the, expression of some genes, but you will have to take a look at each cell, which could be of some number of thousands. So we have to have a much elegant way to be able to understand what kind of different cells that we have in our data set, right? So we are going to use really simple machine learning methods in order to divide these mass cell populations into subpopulations and take a look at the expression of each gene on each of these clusters or subpopulations to understand whether they can be T cells or NK cells or other kind of cells that you're expecting your biological samples to have. Now, that's also a really good way of quality control. Sometimes, different cell types might be mixing some blood cells, maybe mixing if you're using, some, samples from some other part of your body that you don't really want to be represented in your data set, then you can basically cluster, see that, maybe red blood genes are higher in them, and you can just chop them off because they're all clustered, they have a similar expression and so forth. So basically the machine learning concept of data clustering, basically partitioning the data into meaningful subgroups, actually quite nicely apply to single cell analysis as well. And all the steps of analysis that we are doing is to making sure that data clustering is actually working properly. And we're going to talk about what is actually data clustering and how it is applied to our case. So I want you to imagine something, a really, really simple example, right? So I really remember that my PhD advisor used to tell me that what computational science is actually doing, that you're representing a real problem in a mathematical way, right? And you solve the mathematical problem, which you believe that is entailing the solution to the actual problem that you're trying to solve, right? So basically, this actually applies to bulk RNA-seq as well. What we're trying to do is that we have some number of genes that is representing some kind of a sample, and in this case, it's a cell that that you can actually create some number of dimensional space where the number of dimensions will be equal to the number of genes that you have, right? And each of these cells will have a position in this space, right? So I want you to just imagine that we have some, some arbitrary genes, just two, so that we can actually imagine what we are doing here. So let's just say we have gene X and gene Y, we have the expression of a cell of gene Y and gene X, and we have normalized it. Now we can actually see where the position of that cell is. So this itself doesn't actually tell you much, right? So we need a lot of other cells in order to understand what's happening to the cellular population. So the beauty of single cell analysis is that now we have a lot of barcodes and a lot of cells, it's some number of thousands of them, right? And of course, if your library is bad, if your sample is not properly collected, you may have really, terrible number of cells that are high, that have high number of UMIs. But normally, if everything goes good, you will have some quite high number of cells that you can actually see what's happening to each gene in totality. So let's populate this a little bit, right? Let's introduce more cells. So if you do the same thing to all the cells, you may start seeing patterns, right? So this is, again, a really, really easy example. But what's happening is that, as you can see, there are two clusters over here. Although some gene expression of Y and gene expression of X, each cell, is not exactly the same for some, subset of the cells, you can actually see that they are, even though within them, they have some random pattern, you can see that there is a clear division between these two populations, right? So what the analysis that we're trying to do is that we have to capture these clusters. We want to label them, because for us now, in the beginning, there are only cells, right? So what we have to do, we have to incorporate some mathematical method in order to separate them. Now we have separated them, and this will be the case. We'll be having, some label of 1, for example, and some label of 2 for this group on the right. And what we are going to do, we're just going to, incorporate simple statistical tests to see if the gene expression of gene X is higher or lower in both groups. So this is the way we first find subpopulations, and then we are trying to test each gene against these clusters to see what gene is actually making them different, right? So you can do the same thing, to gene Y as well. So basically, when we are trying to annotate our subpopulations, we usually start with trying to look for genes that are representing them. In this case, this population is only represented by gene X plus or something like that. But now that we know that some group has already X, we can just say that this is the only population that Y is substantially high. But again, the choice of label is up to you. Sometimes if we really don't know what kind of a population that we're working with, we just, annotate it as the gene that is up in these subpopulation. But if you're really sure that that subpopulation has CD3, for example, substantially, you can just go ahead and call them T cells. Some subpopulations are obvious, some are not. But basically, we are trying to find those genes that are really high or really low that may actually indicate its phenotype. So what is the problem over here? Yes, David. Oh, Kashi, go ahead.
So when you're labeling the cells in the single cell, it's not automated. It's something for the annotation. You have to do it by yourself. Is
Yes.
that what I am? Okay.
Yes, exactly. So again, I mean, you have to test for the markers. You have to see if the gene is up or down. So it's up to you now. You know, you're making that judgment a little bit. By the way, I'm going to be questioning your biological knowledge as well, and I'm going to ask you guys if what do you believe some subpopulations are based on the data that we see in R. So yes, that is what we are doing. There are some, by the way, guys, there are some algorithms for automatic annotations because, I mean, some cell subtypes are quite obvious. Like maybe if you have a lot of CD3 in some subpopulation, it's obviously some T cell and lymphocytes. But most people want to do this process manually because it's not like some particular gene will always be indicating that subpopulation in every kind of biological sample. So again, it's biology, it's chaotic. So you have to go with your discovery instead of the basic knowledge. But again, you can find, Kashi, other methods out there where you can automatically annotate. Yeah. So, but here, what is the problem over here? What is the problem over here? Who can tell me the main problem over here? This is really obvious. Like I can see that there are 3 populations, maybe 2. What could be the most important, the most simple computational challenge that we may have? Who can tell me? Who can I ask? Alicia, can I ask you? What do you believe the most important challenge over here to find subpopulations?
Maybe I'm choosing the right types of genes to sort by. I'm not sure.
Yeah. Oh, yes, you're really close. But how many genes do we have?
Or we have 2 in this graph, but thousands in cells.
Exactly.
So then which ones? Yeah.
Exactly. So the interesting thing is that I had only 2 genes over here, right? But we have thousands. So we have to actually find these subpopulations in a space that is defined by 30,000 dimensions, right? So the entire single cell workflow, data analysis workflow, is based on lowering this number of genes to some subset, exactly what Alicia said, to choose some genes that are better indications of the cellular populations first. So we're going to do this in a few steps, but I believe we have to take a 10 minutes break, Alper. How do you want to do it? It's 9:52. Is it okay if we take a 10 minutes break, guys?
Sure, let's do it.
Yeah. Okay, so let's come back at 2 minutes after 10, and exactly in 10 minutes. And we'll be back. Okay, it's 2 plus 10. It's already been 10 minutes, so I'll just continue. Let me share my screen. Okay. So in the first hour, we briefly talked about why we are doing clustering, and then we talked about what is the challenge. The challenge is that we have 30,000 genes or features to choose from in order to move on with analysis and make sure that the clustering will actually pick up the cell types that we are looking for. So in order to do that, there are a few steps. It's not like, you can just run one function, it will just do it for you. So in single cell analysis, one of the most important things is that you have to be mindful of each individual step in order to understand that you are actually picking up the right genes, if you're doing the analysis the correct way, and we're going to talk about each of those analyses and the things that you have to be careful of. Of course, this progression in the data analysis is not really linear. It's not like you just do the filtering, normalization, feature selection. We're going to talk about all those and just keep moving forward and do the market analysis and you'll be done. Sometimes at the end of the market analysis, you'll be figuring out something that you've been doing wrong and you want to need to go back to the filtering step. This is actually referring to what you've been asking Nazgul. Let's say that you actually found some cluster whose UMI count are a little bit lower or much lower than you would anticipate. And that may actually mean that those are just some empty cells that you don't want to include in your analysis. So in this case, it might be the case that you will only realize that at the end of the analysis when you do the clustering and find out about the markers of each cell type, and you go back and repeat. So this do repeat process is the case about single cell analysis. And at each of these steps, you may need to go back and redo. So we talked about filtering a little bit, normalization. Now we're gonna start with feature selection. So let me again talk a little bit more about what we've been doing now with principal component analysis and dimensionality reduction. So PCA is considered to be the father of all dimensional reduction algorithms. So why am I talking about dimensionality reduction here? Because we just talked about that we may have 30,000 genes which are equivalent to a space where we have to cluster cells in a 30,000 dimensional space. So that mathematical space should be compressed to some manageable number so that we can trust the analysis that we can do. We can't do clustering on 30,000 genes. I mean, theoretically, maybe we can in some cases, but 30,000 is a really, really huge number. And in data analysis, not just in computational biology, this much of numbers to cluster samples from some number of features, it's actually really, really hard. So we have been doing that with bulk analysis as well. And we basically will do something really similar to single cell too. But we're gonna follow some, quite a number of steps that are kind of expected to be the norms of single cell analysis now. So this is pretty much a really good description on how we reduce the number of genes. We're gonna talk about each of these and the functions in order to do them. So the first thing that we're gonna do, we will choose some genes among the ones that we believe are not really important. Since single cell analysis has a lot of zeros in it, it is highly likely that a lot of genes will have some UMI count of maybe 1, 2, something like that. And we don't really want to incorporate them to our analysis because they are not really changing much. And even though some of those genes may have counts like maybe say 20 or 30, if it doesn't change across all the barcodes that we have, I mean, it's not really indicating any kind of cell type, right? Because it's not really changing. So we don't really need to use that for our analysis because it's not going to contribute to the clustering. As you may remember from the simple example that I've given you up there, the genes usually should entail some kind of a difference between barcodes so that we can pick up clusters. And that's what we are trying to do here. So we are going to choose the variable features first, then be using that 2000 genes. We are going to do some additional, some machine learning based dimensionality reduction to create our own features from the genes. Then we are going to even further reduce that 20 dimensions to 2 dimensions with some of the popular visualization methods like t-SNE and UMAP that you can see in every single cell analysis paper. And we're going to use those two and start interpreting our clusters. So the first objective is to get the data into a manageable size to some 2000 number of genes that we're going to pick our PCA spaces later. So let's go back to this data. So this is what we actually had to start the next session, but you can just skip this because we actually have done that, and we have the number of cells now that we're supposed to have. But you can still use this code to run. It's going to give you the same result. You'll just start from where we are here. It's basically the same object with the same amount of cells after we have cleared it out. So again, Surat is providing a really easy way of choosing these number of genes. And the way to do that is to use the find variable features function. So what find variable feature function is actually doing, that it's incorporating an algorithm to stabilize the variance first in order to choose not the gene that has the highest variance, because the variance of a gene might be affected by the mean expression of that gene as well. So it uses some kind of unique algorithm to stabilize for that variance and tell you the most informative gene from top to bottom. And what we are going to do is that we are going to rank these genes based on their stabilized variances. And then we're going to pick the first 2000 and consider those to be the ones that we need to do our clustering analysis on. So if you click on this link, you can actually find more information about what is a variance stabilization transformation is. This is one of the first papers that came out from the lab that created Surat package. And as you read the paper, you can actually have more idea about some of the methods that are incorporated to do what we are doing today. So let's get started. You can just copy paste this over here and just put it right below the normalized data. Find variable. So one of the things that I really like to do, I like to use comments in before every function that I use or some group of functions that I use, so that if I want to come back, I know exactly what I'm doing. So basically what I'm doing over here is that I tell myself that I'm taking a subset of the PBMC data or visualize all control measures after coming up with new ones. And in this case, after normalization, I tell myself, my future self, that I did some future selection over here. So this, if we can quickly go to the help page of the find variable features, and you can see again, like the normalized data function, you can see all the default values that have been used within this function. As you can see, this part of the package is quite algorithmically advanced, and a lot of these parameters are actually have some default values. So even though you can go with the default values, it may also always be the case that you can take a look at these parameters and you want to change them. And the most important one is that it might be the case that you believe the number of 2000 is not enough to pick the top variable genes for your analysis going forward. What you can do is that you can simply choose this number to 3000. Well, what's going to happen in this case is that you're not going to be choosing the top 2000, but you're going to be choosing the top 3000. And that might be more informative. You might be capturing additional genes that you believe is really important for the analysis. and you may do so. So, but there will be a really nice way of checking that, and I'm just going to be showing you quite shortly. So let's do this normal variable feature analysis really quickly. Again, it's going to stabilize the variances, and it's going to calculate the future variances, and it's going to tell you which of those variables are features. So as you can see now here, before we did the variable feature analysis, we could see that the variable feature count was 0, and we have some 36,000 genes in our analysis, right? So now we have chosen some 2,000 variable features or genes, and overwrote that analysis to the actual object. But if I were to just click on it and enter, now you can see that this number has changed. Meaning is that now there are two sets of genes in the object. We have the original sets of genes that we got from our UME table, and we have the set of genes that we believe that are highly variable, and it's informative for the analysis moving forward. One of the interesting things to be careful about this number is that it is always really useful to check if some of your favorite genes are among the variable features. Let's say that, I mean, it's not going to be in this case, but let me just walk you through there really quickly. So I'm just going to choose assays, RNA. I believe it was here somewhere. Yes. So if you click on meta features, it's going to give you the result of the analysis that allowed you to choose which of these variables were chosen as variable features, but which of them were discarded. So if it's written true over here, this means that this gene was among the first 2,000 to be chosen as the variable features. So the first thing that you can do, you can actually check if some of your favorite genes are among them. So again, this is a table, right? So the next thing that you can do, you don't really have to look for these table in the console over here, but you can view it the same way that we've been viewing the metadata. This is quite different than the metadata, actual metadata itself. This is a metadata about the features. So each feature has some mean, variance, and it's been standardized. And it tells me over here that it was either chosen or not chosen to be a variable feature. So you can just click on any kind of gene that you can think about. Let's say granulysin, which is really important for NK cells and T cells. Not picking up. Okay, because it's not here. I have to find it myself. Should be somewhere around here. I guess I can choose it now. Should be somewhere around here though. Basically, you can actually see if the gene is here or not. In this kind of cases, I have really quick, nice ways of, changing tables. I can actually put this into a really temporary file object. I can change one of the parameters. I can add a new column. Let's say you want to add the row names as a new column to the table by saying row names features. Now I'm going to view temp. And now I have the genes that I now can search them. As you can see, granulysin is over here and it's true, meaning is that it was chosen. So it's a quick way of looking at if your favorite gene is here. Just for to be sure that you're doing correctly.
So that search function cannot search through that first column? Is that what?
Yeah, it does not. Yes, yeah.
Oh, okay.
Yeah. I just figured it out now for some reason. Yep. There are different kind of tables where the gene column is there or not. So you can manipulate the data a little bit as I've been doing really fast, and just take a look at the table really quickly. Let me just share this with you as well. No, that's why R is really good. It's not a just 100% interactive package. You can just manipulate the data a little bit to get more out of your tables. Okay. But what you can do, you can take a look at the genes in a different way. You can pick up some top 20 of them. There's another function for that called variable features. It basically gives the entire list of features for you, which is of length 2000, right? So what you can do now, it's already sorted. You can pick up the first 20. What I'm doing over here is that I am searching for the top 20 elements in this list, which has the all 2000 variable features that we've been searching for in the object. It's going to give me the top 20. I put that in top 20 object, then I'm just gonna, print that, and there you go. You can look at the top variable features over here as like this too. So we have granulysin over here. Can anybody tell me if they're recognizing other genes over here that could be found in a cell type in a PBMC data? Who can tell me?
NKG7.
NKG7, yes. Kashi, we love NKG7, aren't we? Okay. Yeah.
Maybe granzyme B2, things like that.
Yeah. Is anybody recognizing this? So we're going to have a small amount of cells that have this gene, but it's highly variable, even though it's just expressed in a few number of them. It's going to be a gene that is expressed by platelets in a PBMC data, but we're going to shortly see that that those genes will only be found in those cell types, and the cluster analysis will be able to pick up those cells that only have this gene as well. So now you can actually visualize this, right? You can see, you can just plot this really quickly, the mean expression and the variance of each gene in the plot, right? And Surat also allows you to put the names of those genes on some top value with additional functions. I am not sure why. Okay, because I didn't run this. So basically what I'm doing over here is that I'm not just plotting the plot over here on the right, but I'm actually putting its returning value to an object. You can do this in R as well. So all figures, you can actually put that in another object as a different name. Just click on it, and we are going to use this to add the text on it again. Then we're going to plot. And we can actually see which of those highly variable genes are where and what are those variances over here too. This step is really delicate. You have to be choosing your genes really appropriately here. Sometimes when we have multiple cell types that we want to further investigate their subpopulations, let's say when we have T cells, we know that some clusters are T cells. Now we are separating them, and we redo the analysis in order to choose some 2000 number of genes that are specific to T cells in order to move forward. You have a question? Yes, but here's the thing. In order to make DEG, you have to have clusters first, Nazgul. So the reason that we are selecting these genes is not that we are discarding them. We are just trying to find genes that are indicative of the clusters. So yes, I mean, it is highly likely that if you pick up some top variable genes like this, right? So it is always possible that you may see some other genes that you didn't pick up as being high or low in some of those clusters, but having too many clusters will damage your clustering analysis. So what we will be doing is that we're going to be testing all these genes, but what we're just using these top variable genes for clustering purposes. So the marker analysis, or you can call it differential expression analysis too, in this case, is just independent of that process. So everything that we do here, we are doing before clustering. Does that answer your question? All right, so now we have some 2000 genes, right? So what is the problem? The number is still so high, right? I mean, those 2000 genes are really important. That we know. It's okay, right? But let's take a look at this in a different way. So all genes other than those 2000s were not really changing much across the barcodes. Their UMIs were either too low or too high, but they didn't have much variance. But now we have a lot of genes that have a lot of variance, but some of them are redundant. Why is that, right? Because sometimes you have multiple genes that are giving you the same indications of the same phenotype, right? So basically you may have one gene that is correlated to another gene, and we know that it's not like, all genes are independent, and a lot of them are actually correlated. So what we're trying to see is that if there are some correlated genes among these 2000 selected ones, we're trying to, in a way, come up with a lower number of some kind of genes. They're not going to be actual genes, but we're trying to find some lower dimensional space that will be still representing the information that we have from 2000 genes, but we wouldn't have 2000 dimensions to start with. So who knows what curse of dimensionality is here? Have you ever heard a term like that? Maybe I could ask this to Bruce, because I heard Alper mentioning that Bruce was a statistician. Bruce?
Here we go. Excuse me. Yeah, could you repeat that? Trying to do email at the same
So
time.
you know what curse of dimensionality is, right?
Yeah.
Yes. Okay. So the curse of dimensionality is that when you have too many dimensions in your data set, too many variables in your data set, even though you have some pattern in there, the distance between the points in that two-dimensional space will be so high that the closest distance and the highest distance will be equal. So it will be really, really hard to pick up what is close and what is not, which is really essential for clustering analysis.
Right.
And we'll
I mean,
see,
it's a question of the forest and the trees. If you've got too much dimensionality, you lose track of what's going on. And you're right, statistically, it's hard to pull them apart at that point.
exactly. And this is the next step, even though, as Bruce was saying, that we have a lot of genes and this could be useful, but we can literally pick up some of those genes that are actually behaving similarly, and we can just represent them with just one variable instead of some 20, where all those 20 genes are correlated with each other. So there's a really great explanation over here to what's going on. Just imagine that, just forget about the fact that we have genes now. Let's say that we have just two dimensions. As you can see, even though there is some random pattern, the first variable and the second variable, they are actually correlated. So as one is increasing, the other is increasing as well. So we don't really need either the first one and the second one if we have the other, right? So what we can do, of course, in this case, we can just choose one, but maybe we can just combine them in such a way that, and we can create another variable. In mathematics and statistics, is called the latent variable, a variable that does not exist, but is created from the existing variables. You can literally come up with a linear combination of these two and other different combinations that are independent from each other. So this vector is actually representing that the angle between two vectors are 90 degrees, which is indicating that they are actually picking up some combinations of genes or variables in your data, but they are independent from each other, which is also a great, feature that you want to have for your clustering, when the variables that you use for clustering are independent from each other. So basically what we want to do, we want to find these kind of combinations. And if you actually find that combination, for each of these samples over here, you can actually turn this data into what you can see on the right side, right? So again, there is some variance for the second principal component. Basically, the name principal component analysis, the dimensional reduction that is applied over here is actually referring to the fact that you are capturing those components that are principal to your data. So that's why it's called principal component analysis, and everybody's calling it PCA. So basically what's happening is that you have the first component and you have the second component. Both are some combinations of the variables that you have. But as you can see, the variance of the second component is quite low, which may actually tell you that you can ignore it. So in a way, you actually have the information of both variables over here, because the first principal component is a combination of your variables, in this case, genes. So in a way, with just one variable, you can represent all other variables that you believe that are correlated to each other in just one go. So this is a really great feature of PCA. It's been around for like many years, and it is in most places are regarded as the father of all dimensional reduction analysis methods. And this is the basic first principle, the dimensional reduction method that is incorporated in Surat. So the first thing that we're going to do, let's go back to the R again. So now we have chosen our variable features. Now we're going to reduce the dimensionality. Just a formality, before moving on with PCA, you have to further scale your data as well. So we quickly run this code here. And after that, we are going to run PCA. Again, we are going to input the object name. Verbose false means that you don't really want to have information about the progression of the analysis. You just want it to be done. So basically, if you say true, it's going to print something that is indicating that it's centering and scaling the data. But when you do false, that information will not be printed, even though it's doing the same thing. So then we're going to move on with PCA analysis. It's just a really simple code. You can just say run PCA, the name of the object, verbose false, and you have to choose the number of pieces that you want to extract. So basically, what we really want to do is that we don't want to use all those 2,000 genes, but we want to use some, 30 combinations of 2,000 genes that are more informative than other combinations. So basically, then you have 2,000 variables over here. You're going to have the same amount of principal components, like 2,000. And which will be the case theoretically is that some number of those with the highest variance are the most informative. And after some point, you don't really have to use them. You can just pick up some first 20 or 30 and whatever the number. So let's quickly run this again. And let's look at the data as well. See, so the printed message actually changed a little bit. Now we don't have 0 variable features, but below we have an indication that we have done some dimensional reduction to Surat analysis, and it's been recorded. It tells you that there is some principal component analysis have been conducted for this data, and it's coded as PCA. You can actually give a name to these as well, because normally when you do more analysis, you may have several different dimensional reductions, and you can give a name to it. But if you don't, it's just going to call it PCA in short. And we're going to have more dimensional reduction methods now that we're going to apply, but they all will have a different name. Elbow plot. So you may pick some first 30 principal components, but you may still not know if you really want to use that first 30 or not. So there's a basic plot it's been around since principal component analysis was first started being used, and it's this one. So again, Surat is allowing you to use some of the basic machine learning algorithms and methods to visualize them with really easy commands. The only thing that you have to do just to run elbow plot right after PCA. So if you do that, it's going to visualize the variance of each principal component in a ranked order so that you can actually pick the number over here. So as you can see, the x-axis over here are the index of the PC, and they are sorted. So the first PC will have the highest information, the second will be the second highest information, and so forth. And the y-axis will be indicating the standard deviation of each of those PCAs. And it will actually tell you how is the amount of information changing as you add more principal components to your analysis. So basically, we are trying to pick the actual number of PCs that we want to move forward with clustering later. So the rule is that you are trying to find some kind of an elbow. That's why the plot is called elbow plot, where you see that the more you add, you're not gaining more anymore. So basically, as we go through the first, the second, the third, the fourth, the fifth, until the 10th and the 20th, maybe after 20, I don't really need to, pick principal components anymore. Even though I ask the run PCA code and Surat to generate me the first 30 principal components that have the highest variance, it might be the case that you don't really need the first 30, but you may actually do it with the first 20. Again, what we are trying to achieve with clustering analysis is that we want to pick as much variables as we can without choosing a lot more that will hinder our clustering analysis and pollute the clustering analysis and will give us wrong cell types. So what I'm understanding over here, and it might be different for you guys, you may just say, okay, yeah.
Sorry, I stopped you, but I got confused here. If we are using all the 20 genes for the 20,000 genes for the PCA, then what's the role of the variances that we found?
So the variance that we found over here, meaning is that the first principal component is actually picking up the genes among the 2000 that has the most variable ones. And most of these are independent. Basically, each of these principal components will be representing some different part of the correlated genes in the list of 2000. Let's say that you have some 20 or 30 genes that are correlated to each other that has the most information. That is going to be picked up by the first principal component. So basically, you have the same amount of information in one variable instead of using 30.
Yeah, so I meant about those like 20 genes that we picked earlier as variable features.
Mm-hmm.
So they are used, their deviation is used for the PCA, or
Yes. Basically, we have the same or manageable side of the variance that is represented by 2000 genes, but we collapsed it to some 20 new variables.
I see. Okay.
Yeah. So that is the basic ideology of dimensionality reduction analysis, regardless of the fact that we are doing a single cell analysis now.
Right.
You can see that the same application is done everywhere.
I have a quick question too. So each PCA represents a comparison between two genes, is that correct?
No. It's some number of genes. So it's a combination of some 2000, but that example was just a representative. So this analysis is just an illustration in 2 dimensions, but we are actually doing the same thing in a 2-dimensional space. 2000 dimensional space, I'm sorry.
So each PCA that we did, will it take each gene and compare it to all the other genes from the 2000 or?
Yes, so you in a way, I mean, it's roughly, you can say that you're comparing each gene to all other genes. You're basically calculating the correlation between all those genes, and you do some linear algebra to come up with a different data set that only has 20 dimensions instead of 2000. The theory is quite complicated in a biological sense, but basically it's actually trying to figure out which of those genes are highly correlated to each other, and it's compressing those to just one dimension instead of using all those correlated ones.
And then you pick the top 30, the ones that have the least correlation between each other. Is that correct?
No, not least correlation. So each of these PCs are representing some high number of genes that are correlated to each other. So basically they have the same kind of behavior. As one increases, the other increases as well. And it's just picking those up.
I thought we wanted to discard, just pick one basically from a group of genes that have the same variability or variance. We want to just pick one sort of like as a representative.
No,
Or-
so basically the first step was we discarded those ones that, regardless of whether they were correlated to something else, didn't have any information. Now we have a lot of genes that have a lot of information, but we are just technically lowering the dimensions without losing any information about the variance. So basically the distance between the relationship between the cells will be the same, but we'll be dealing with 20 dimensions instead of 2000. So basically just it's the compressed way of looking at the data without losing much information. But we are just showing how to pick those and how to interpret the plot to pick the number. So again, you can take a look at this plot and try to figure out the number of dimensions over here. And I'm just going to reduce this to 20 dimensions moving forward. But some other people can just say that, okay, so this is enough variance to me, and I'm not going to move forward. And this is the only way to figure that out is to cluster the data and see that if some cell type you believe it will not be represented, and you can go back, maybe introduce some more PCs by increasing the number of pieces that you pick up and move forward. So you can also draw a heat map of these to see what kind of combinations of genes that we have. So in this case, I'm actually visualizing the relationship between the genes and the principal components of the first 6 PCs. So it's going to tell me that these genes have in some way they are correlated and they are represented by PC. And there's another combination over here, another combination over here. Basically, we have information from a lot of those 2000 genes in these principal components, but we don't really have to use the entire 2000 genes data. We kind of compress them into 20 dimensions. The theory in this is a little bit could be a little bit complicated, but basically just imagine that we are compressing our data so that it can be manageable for clustering. So now you can actually visualize your cells in this new dimensionality as well. You can use the function dimplot and indicate that you want to use the reduction PCA to visualize the cells of your object. As you can see, we start seeing some clustering pattern, right? So there are some PCs that are related to some genes, and it starts giving you some patterns. But the interesting thing will be here is that we still have 18 more. Oh no, maybe I just didn't really pick the right ones. Oh, no. Okay. So we still have 18 more principal components. They may actually indicate additional variation over here. So here you can see that you have 3 clusters, right? But I haven't still, looked at maybe the 3rd principal component and the 4th, and I have, 16 more. So maybe there are some other clusters over here that I can't really see just on the perspective of 2 dimensions, right? So I need an additional way of looking at the data in a 2-dimensional perspective, and I'm going to use additional dimensional reduction methods that I reduce those 20 dimensions to even further to 2 dimensions. So I just want to take you back to this initial plot over here. So as I said, so the dimensional reduction routine of single cell analysis is quite rigorous. And you have to actually follow these steps by choosing the first variable features, then reduce them to PCA. And the reason that we don't really go directly from variable features to t-SNE and UMAP is that we're going to do the clustering on the PCA, but we're going to visualize this on TISNE or UMAP. So there are other applications of TISNE or UMAP where you can actually cluster directly in the okay. What's up? So our director is here. Thank you. Interrupting me. Okay. So
Is that for here?
yes, continue. Hold on. So basically, you can actually do the clustering on TISNE or UMAP as well. Some people are doing that. But what Surat is actually doing that is that you reduce the number of variable features to some 20, 30 PCs, do the clustering on PCA, but visualizes on TISNE and UMAP. So for Surat, in this case, it is much better to visualize the clusters in TISNE and UMAP, but it's better to cluster on PCA. In another package, you may find another method, but this is the way Surat is going. So let's quickly move forward. So I'm just going to visualize TISNEs and UMAPs. So again, going back to the elbow plot, we decided that we're going to choose 20. Of course, this number could be different for you. The function again is quite simple. You run TISNE. Now you can take a look at the object as well, print it. Now the number of reductions actually increased to 2, and the names are actually given over here. And you can use the same function, dimplot, by just replace PCA with TISNE to visualize the TISNE as well. This is a really great way of looking at the plot. It actually beautifully reveals how many clusters that you may have. So I'm asking you, any one of you, how many clusters do you see? Who can tell me? Let's make a competition. Let's try to see who's going to guess.
12?
Let's count, maybe. 1,
I'd say
2,
6.
6? Okay. 1, 2, 3, 4, 5, maybe. Do you think this is one cluster or two?
I think it's 2.
I
It's
think
two?
1.
You think it's one?
Yeah,
Yeah, okay. Who's saying that? Bruce?
yeah.
Okay. So yes, I mean, the definition of cluster is really interesting here. So the cluster is basically a group of cells that are closer to each other, right? But to what perspective? So I'm going to disagree with Bruce here. We're going to see that there are some clusters in it too, but it's basically depending on what perspective you're looking at.
Right.
So Simona is saying to 11. Okay. Simona might be right, basically, but I don't know which clusters that she was thinking of that we found over here. So this is one way of looking at possible clustering, patterns in the data. But you can alternatively use another dimensionality reduction method too. Its name is really simple. It's UMAP instead of TISNE. I mean, you can try reading the paper of both. I actually may have put some, some YouTube videos and papers over here for you to look. But its theory, it's much more complex than PCA. So the only thing that you need to know about TISNEs is that if two cells are so similar to each other, they will probably be in the same cluster that is visualized over here. But again, that information might be missing some of the information that is covered by 20 dimensions, which Surat is using for clustering. But some people will disagree and will just say, okay, I mean, this is a cluster, so I don't really need PCA. I can just pick this up as a cluster as well. And that's what we're going to see too. So you can just use UMAP as well. It's an alternative way of visualizing the clustering pattern. Again, you're picking up some clusters. You can see that maybe this is a cluster, this is a cluster. This could be a cluster, by the way, because it has some distinctive shape as opposed to this main cell island that we have. This could be a cluster and that could be a cluster. the theory behind two of these visualization methods to two dimensions are different, but they are basically showing the same thing. You know, they are revealing the possible clusters over here. So we have reduced the data. Now it's a manageable size. We picked genes, we picked PCAs. Now what we have to do, we actually have to do the clustering. So this is the most important analysis in the entire single cell analysis workflow. And everything that we have done so far to make the clustering patterns look more manageable and allowing the algorithm to better understand which clusters that we're looking for. Basically, data clustering is defined as partitioning samples in a data set into meaningful groups. And what is a meaningful group over here? Basically, we just covered that. Each dimension in a data clustering analysis is actually representing a gene expression or some transformed gene expression that representing the expression, all the other genes that are correlated with that, right? So what we are expecting is that if we have done a really good job reducing the dimensionality of this data, the distance between two cells that have the same cell state or cell type will be so close to each other, as opposed to cells do not have the same phenotype or the expression profile, which will be really far away from each other. So when you have some points far away from each other and some that are really close, you can actually apply clustering algorithm to easily pick up those clusters that you believe are representing cell types or cell states that you are looking for. Obviously, this is a really, really easy example, again, in 2 dimensions, and we are doing this in 20 or 30 dimensions and whatever the number of pieces that you picked up over here. But what we actually gonna do is that we will try to even cluster the most absurd data sets that you may not be able to, you know, pick up easily. So who can tell me how many clusters that you see over here? Kashi,
Yeah,
What
I don't know.
cluster?
Maybe 3, but probably more.
3 is a really good number, Kashi. I was actually surprised that you would say that.
Yeah, no, I think I see 3, but I don't know.
Nazgul said 2. It could easily be 2, by the way, Nazgul, by the way, because it's really hard to pick up, right? You know, it's not like you're always going to have these clustering patterns that are really, really easy to pick up. But, you know, if you really want to pick some number of clustering, you would probably pick this as a cluster. This is a cluster. And obviously, these cells are more closer to each other than all other cells, right? So this is what we are trying to do with data clustering and try to understand what is the optimum, not the actual, the optimum number of clusters that you can easily pick the differences among them. So let's give a 10 minutes break and we're gonna come back to clustering and start making marker analysis and search for cell types and cell states. So let's say 10 minutes and come back at 11. Does that sound good? Let's come back at 11 then. Okay. It's 11 and I think I'll start now. Tomoya has a question. The distance of each dot means similar to the gene expression of the cell on t-SNE or UMAP plot. Both actually, Tomoya. The only significant difference is that UMAP is doing a little bit better job than picking up the distances that are far away from each other. But both t-SNE and UMAP actually tells you if those two cells are closer to each other on the PC space, they are closer to each other in the t-SNE and UMAP space as well. So the difference between UMAP and t-SNE is how accurately they are picking up the distances that are far away from each other, other than the closest ones.
Thank you.
Does that answer your question? Yes, okay.
Yes, thank you.
So start the presentation now again. All right. Now, so let's start clustering, right? So basically, guys, clustering is not an easy problem. There are maybe thousands of clustering algorithms everywhere. So this is some people's job. They come up with new algorithms. They try to see if their algorithm is better than for some other people's algorithm into some perspective. But there are basically some clustering algorithms that are believed to be working perfectly for certain applications. And one of this is specific for single cell analysis, and that is a graph-based methodology. So basically, the first thing that you do first is that you look for cells that are closer to each other and you establish connections between them. So imagine that these two points as cells, and if they are neighbors to each other, you basically draw a line between them. So what is the criteria for having a connection from one cell to the other? It is that this connection is actually to a cell that is the second or the third closest value. Let's say that we call these k-nearest neighbor graph. If you have a k-nearest neighbor graph, each point will have three lines or edges to the first three cells that are closest to it. So you basically try to have as much connections as you have possible to cells that are closer to each other. So one thing that you can pick up actually is that since these group of cells over here on the top left, they are so close to each other, the majority of the neighbors of each other are themselves, right? So this that you can see that this group of cells has a lot of connections to each other, but not much connections to the other part of the data, right? So we are going to use these graph system in order to partition the data into meaningful groups. You don't really have to do this all the time, but that is the clustering methodology that is followed in this particular package. So the first thing that we're going to do after finding the PCs and doing all the dimensional reduction things, we are going to move forward with finding these neighbors, right? So what's going to do is that it's going to, look for the closest cell from each cell and build connections between them and build a graph. So basically, we won't have an expression profile anymore, but we will have just some, cells that are referred to as nodes and connections between them. And this will be the data that we're going to use for clustering. But we'll be still visualizing the relationship on the reduced dimensionality. So you can just pick this up. We are going to use the first 20 PCs dimensions, and we can go ahead and say, just click. Say it just said that it computed the graph. It comes up with another graph on top of the nearest neighbor graph that we just talked about. I'm not going to go into too much detail because at this point we have to start making biological interpretation based on the clusters that we have found, but make sure that if two cells are closer to each other, they are going to probably have an edge or a line between them. And that information will be used for clustering. So you can take a look at these object. You can just say neighbors here. Oh, no, it's not here. So I have to go to the essays. Should be somewhere here. Not yet, maybe. I was pretty sure it was here. Yeah, the graph, yes. So it actually tells you that it has some distance between them and the neighbors are found. So it keeps populating the data as you have more graphs and more data in it. Now, what is the indication? What is the reason that we are actually using these edges? So basically, the thing is that if you have a bunch of cells that are closer to each other, they are going to be neighbors of each other. And these neighbors will be picked up by these edges because they are actually using the information that each cell is a neighbor of each other, the other, right? So you may have still some lines going from one cell to the other, which may not be in the same cell type or group, right? But basically, the majority of the lines will be drawn within the same group that are likely to be from the same cell type. And that is the algorithmic, conceptual definition of what we are doing with finding clusters. But the interesting thing is that how do we determine this threshold of having a lot of lines between these cells, right? So we have to have some kind of a parameter or some kind of a threshold to determine what is the optimum amount of lines that you have in a group of cells in order to consider them a cell group, right? So that may actually be depending on the situation, and that is actually giving us flexibility to play with our data a little bit and pick apart cell types. Because let's say that you have some T cells. Of course, T cells will be so closer to each other. But the problem is that you may have some additional cell types within T cells, like effector T cells and naive T cells. And even though they have some signal that they're indicating that they are lymphocytes or T cells, they are going to have some specific markers specific to them as well in order to pick them up. But it is our job to play with the parameter and keep discovering these cell types. So the parameter, in order to define the number of clusters that you're going to have, is the resolution parameter. And this is the main parameter that you have to keep playing with in order to understand how many clusters do you have. So I'm just going to run this really quickly. So what it did was that it ran the algorithm and it found some clusters. And where these clusters are stored, you can just take a look at the metadata. Right? So as you can see, each partitioning is coded as the graph that you have found, which is the graph based on the RNA assay. In this case, Surat will allow you to do multiple clustering runs on multiple assays. So the notation that you have over here is that this partitioning is based on RNA. It's based on the nearest neighbor graph that was based on RNA, and it's using the resolution parameter 0.2. So in that way, it's going to start labeling clusters starting from 0 to some number of clusters that you have found. And it's also going to store the last clustering run that you have as another label as Surat cluster as well. And in this case, we only had one clustering run. So that's why the labels of them are actually identical. But as we'll see, we're going to use more resolutions later, and we'll have more, and the Surat clusters will always be equal to the last clustering that was computed. Anybody have any questions until now?
So, do you determine how many clusters it should find for you?
Yes, Kashi, it's a iterative process. What we actually have to do, you have to keep dividing, keep searching for markers. make sure that those markers are legit. Maybe you don't really need those markers. Sometimes you may actually picking up two clusters that have two different markers, but they may not be really useful for the analysis that you're conducting.
So you just like play with the numbers, say, okay, find 3 clusters, and then if that makes sense, you go with it. If not, you make 4 clusters.
Exactly. Exactly. Yes. So it's a treasure hunting process. You just have to keep playing and keep searching and keep analyzing the data. At some point, you may need to want to go back, believing that you haven't done the normalization on the filtering properly, or you may not even chose the right amount of variable features or the PC. So you may really need to go back and start from the beginning. So that's why single cell analysis is a little bit more complex than bulk RNA-seq based differential expression analysis.
Right. So it's more of a like, there is no right answer that how many clusters are correct. So it's just more of a manual job in some sense.
Could you repeat that again? I'm sorry, Kash.
So there is no like correct answer on how many clusters is exactly correct. It's more of a manual job. So it's like some sort of like, arbitrary choosing, but you have to like validate with further samples or something like that.
Exactly. I mean, but I may actually provide you in a good, process for determining a good number of clusters. But that will only be possible by visualizing multiple resolutions in the same time. So Nazgul, going to your question, supervised clustering is a little bit more advanced than the unsupervised clustering algorithms that we talk about here. Basically here, the clustering is unsupervised. But the supervision meaning is that you already have some information about possible clustering that you may have. We are conducting some kind of a project like that as well. We devise supervised clustering, but it's a little bit more advanced and you have to know what you're looking for. Basically, let's say that you are definitely sure that you have T cells in your clustering algorithm. You can, in your data, you can literally, input, a CD3 parameter to make the clustering algorithms to search for clusters that only have CD3. So that is possible, but I don't suggest it all the time. You have to start with unsupervised clustering to have an unbiased opinion of the different, populations you may have in your data. So in your case, I'll start with unsupervised, and maybe if I need it, I would use a supervised, version as well. But the most clustering algorithms are unsupervised. They are the most basic ones. So what happened when we picked resolution 0.2? So I did that, and I am going to visualize this. Okay, question. Is this number of clusters enough? Who can tell me? Let me ask someone else. Simona, are you there?
Yes.
Okay, how many, do you think that we did a good job by separating the data?
I don't know. Since you say also that the cluster can be distinguished by shape, right?
Yes, we can say that. Yes, I mean, even though this is a
looked by me that, for example, I don't know. You of course you can't see my arrow, but it looks like the red one has two shape as well. The green has two shape.
yeah,
For example,
exactly.
the
Go ahead.
yes, exactly.
Yeah, so yes,
But
I mean,
probably I'm wrong.
no, you're actually right. So again, yeah. So again, I mean, that could be interpretable, Nazgul. It might be the case that UMAP is better in some cases, but there will be possibly easy way to determine if you haven't necessarily cluster enough. But yes, I mean, Sandhya is right. We may actually need to cluster further, but this can be picked up from Tisney as well. But yes, in some cases, Nazgul, you're right. UMAP might be a little bit better to pick up, but Tisney actually works fine in this case too. So we have to cluster further, right? In order to do that, we're going to increase the resolution parameter. But I actually like to do a different approach, which is the method that I've been referring to when we were talking to Kashi. Instead of going one by one, and this I know because I worked on clustering a lot, especially during my PhD and in my lifetime, is that it's not enough to just look at one parameter. You have to take a look at several parameters in the same time to understand how clusters are divided as you increase the number of clusters that are picked up, right? So it is better to take a look at multiple answers in the same time in order to understand if those clusters are separated just randomly, or there is some kind of a pattern that you that you missed just because you didn't increase the resolution enough. So the meaning is that theoretically you can keep increasing the resolution and keep finding new clusters. But the idea will be is that if you keep increasing the resolution and you keep seeing the same number of clusters in some clustering results, those clusters will be the legit clusters that have a lot of signals specific to them. So basically what's happening over here is that the resolution parameter, you can just change it with a vector, and that vector may have multiple parameters in the same time. And you can just run it. What it's going to do, it's going to partition the data 1, 2, 3, 4, 5, and 6 times in different ways, and it's going to give you the labels associated to each of those clustering analysis. So let's do that. See, just repeating the same thing each time. It tells me how many clusters that I found. So in this one, it was 6. It was the exact number over here because we started with 0.2. Then we increased that to 0.4. This time it found 9. Then it was 0.6, 11, 11, 12, 12. So maybe we can kind of understand that there's a pattern with all of this. And there's a really quick, function here that you can use to combine all different results in the same time so that you can take a look. So basically, each time what I'm doing is that I'm selecting t-SNE reduction. I'm labeling each cluster which will be either 0, 1, 2, or 3 based on each individual number of clusters that you have in each clustering algorithm. And you're going to group these labels based on the labels that are stored in the metadata. So these are actually coming from the metadata itself. I can quickly take a look at it. As you can see, we have separate clusterings for each of them, right? Starting from 0.2, 0.4, 0.6. They are all based on the same graph and the same assay, but we did just with different resolutions. And the last resolution that we have used is again stored as Seurat clusters. So you can literally pick those up by introducing another parameter to dimplot function, store them in individual objects in R, and by adding all of those to each other, you can actually visualize this in one go. This is now looking much better, don't you think? So you have multiple results, right? I mean, you have different perspectives to choose from. For example, when I asked Simona told me that I would probably pick this cluster apart into 2, which basically is happening is I increase the resolution further, right? So I have 4, 5 over here, 3, 5 over here. Their labels are different. They are different numbers, but basically they are the same cluster. So the label will be different, but the partitioning will be the same. So basically what I'm seeing over here is that this one actually was separated into 2, but nobody actually told me about the small cluster over here, which was considered as a part of this cluster 1, but it was so close to these 2 other clusters, it was considered as a part of these cluster, but it was apparent in t-SNE that it could be a different cluster, and it's actually picked up as I increase the resolution.
So the correct one is 11 or 5 or 6, sorry. What's the correct one?
It's not, there is no correct way of doing this, by the way, Simona. It's again, based on your judgment. So we kind of have an indication that it could be 0.6. That's what I'm saying,
see.
because that pattern is not really changing much if I keep increasing, right? So there is a thing that here you have two clusters over here, but this division of 0, which was initially from 0.8, could be arbitrary. I don't really know. So it's basically after I analyze 0.6, I can go back and analyze 1 as well, which might be the case.
Is there a reason why there's random ones in clusters? Like in the second graph, there's a pink in the green cluster?
Oh,
Yeah.
yes, this is a really great question. So clustering algorithms are not perfect. Sometimes some points are, really close to both clusters, right? But you have to decide whether it's on the right side or the left side. So basically, since we are trying to find different patterns of cells having different markers, it is okay to have 1 or 2 cells that potentially could be misclassified. So in this case, you don't really have to worry about that. The only thing that we have to understand is that the majority of the cells that are labeled as such are actually looking much more closer to each other, because we're trying to understand patterns over here.
Thank you.
Okay. It sounds quite simple, eh? So that's the thing. But if, again, I mean, to answer Simona's question, there is no right or wrong answer here. So you're just discovering. If it was just one, linear workflow, you wouldn't be doing your job. Sometimes, as we'll see, this clusters 1, 2, 8, 6, and 4, they are actually T cells that we're going to pick apart as CD4s and CD8s. And what's going to happen is that maybe you just want to, isolate them, create another Surat object just by choosing them, and redo the analysis to see if you're picking up additional clusters. It's all about zooming in, searching for new patterns, keep clustering, and try to understand if you can find different cells with different markers in them. More like a, iterative process than a linear one. So in this case, I mean, to go with the plan, I'm going to use 0.6, but again, it's up to you. Now, the fun part. Okay, now we know the clusters, but these are clusters based on PCs, right? The collapsed data that we used in order to reduce the dimensionality from 2020. But now the question is, which of those genes are making these clusters unique, right? So basically the PCs, the principal components, are correlated to genes, and because of the, high expression of those genes, we actually have these clusters, right? They are correlated. So what we have to do over here is that we have to analyze each cluster at a time and check if each gene, whether it's up in one cluster as opposed to all others. So this is a really, really fast algorithm and really quick to use. By the way, were all of you guys able to write the codes that I did, and you are where I am now? I haven't heard any questions, so I believe that you are following me. So the one thing that we have to do in order to test for the clusters that we have found in resolution 0.6 was that we have to set that as an identification. So you're using the function idents and the name of the Surat object to say that you will have to do all the analysis based on this partitioning. So when I do this, whenever I visualize something or do some kind of a testing, this will be the set of clusters that I'm going to work with. So when you click on find all markers, what it's going to do is that it's going to test each gene for each clusters against all of them. Basically, let's say that you are trying to see if granulysin is an NK T cell, an NK cell or T cell, cytotoxic T cell marker, is that you're trying to see if one has upregulated or downregulated relationship with one, meaning is that this gene is up in one as opposed to all other clusters in the data set to see if granulysin is up in one and not up on the others. So let's quickly run this, and it's going to take some time. It's going to take around maybe 1 minute, even though this is a really small sample with just 1,000 cells. It's just going to take each cluster at a time and test each gene for each cluster as opposed to all other clusters. At some point, we will see that some clusters will be harder to determine their annotation or their cell type identity. In this case, we are going to do a bit more focused analysis, and where we are going to compare cluster 0, not with all other clusters, maybe just cluster 1 against cluster 2, which will be the case over here. So as this is going on, let me just tell you that. See, so 0 is quite far away from 1 and 2, right? But 1 and 2 could have easily be mistaken as 2 separate clusters, but we were able to find them. So when we do a statistical analysis testing in order to understand what makes 1 a separate cluster as opposed to all others, or as opposed to 2, we may actually find similar markers in both 1 and 2 because they are really, really close. As you may remember, the distance actually representing how similar the expression profiles are. So it meaning is that if the clusters are closer to each other in t-SNE, it is a good indication that they are actually similar to each other as opposed to all other clusters. And going back to your question, Nazgul, if 2 clusters are really close to each other in t-SNE, it might be not be able to pick up as good as in UMAP. So UMAP is actually better to understand if 2 clusters are closer to each other or not. But in this case, we were able to see that 1 and 2 are different, and we're going to test that as well. As I've been describing this to you, the analysis is nearly done. We had 11 clusters, and for each cluster, we analyzed each gene and calculate log-fold changes and adjusted p-values to see if that log-fold change is high and it's significant to understand the markers. Okay. So the result will be a table. So that's why I didn't actually write it on the Surat object itself, but I wrote it on a table. If you just click on it and just place enter, you'll see that it's a really simple table where you have each gene as a row name, and it gives its log-fold change, p-adjusted values, and some other measures, and it says that it's specific to cluster 0 and the gene as well again over here, but we have all the clusters here too. But the best way to look this table up is to view it. And this will be the table that you have to search for potential markers across all the clusters. So somebody give me a gene name that could be important for PBMCs to pick up some existing cell types in it.
CD14.
Could you type that down to Zoom or Slack? Okay. Yeah. So as I believe, there are some cell types in the PBMCs that could be determined by CD14 signal, and those could be probably the CD14 plus monocytes. Am I correct, Alicia?
Yes.
Okay. So it says that log-fold change of CD14 is positive, meaning is that as opposed to all the combination of all other clusters, the mean expression and compared to the mean expression of 0, cluster 0, the log-fold change is around 3, which is quite high. And the other thing that you may have to look for is these 2 measures over here, which is indicating the number of counts in each cluster that has non-zero for this gene, meaning is that more than 90% of all these cells in these clusters, even though they are similar to other cells in the same cluster, as given the expression profile, it has this gene up, not zero, almost more than 90% of it. So it's a very good indication that the phenotype of these cluster is determined by CD14, right? So to quickly take a look at all these, you can do different kind of things. So this is not a linear process. You may actually need to search for multiple, genes in the same time. For example, you can search for granulysin again. And in this case, you will see that granulysin is up in both, although in one, it's really high, but in the second cluster, which is cluster 6, it's somewhat high. It's again high, it's significant, but not as high as cluster 7. So this will probably be the NK cells that we have, and the cluster 6, which is over here, will probably some CD8 subtype that is cytotoxic. So let's move on, but we need to have some different ways to look at these relationships in different ways. This is one way. You can also, what you can also do is that you can use additional packages to marker heat map to pick up the top some top number of genes differentially expressed in each cluster with giving some number, right? Maybe 10, maybe this could be 12 as well. And you can literally pick up all those genes that were significant and visualize this to create a heat map. So what this is actually doing is that it searches for the genes of each cluster in this initial table that we have. Then it groups by cluster, which will do some kind of a ranking in each of them. It's going to use the function top n to search for the top number of some entries when they were ranked by average log-fold change, which is the thing that we are looking for. So if you do that, you can simply run this. Oh no, I just have to. What's happening? Could not find function. Oh yes, I have to use this library. Not the other. Sorry. Just gonna put this in here over here on Slack. You have to call for the deep layer library. There you go. Yes. So this is actually a reduced version of the initial table that we have. Instead of all the markers, it has all the top markers in that table. So that you can see that each of those genes are actually up in each of them. So for example, let me see this gene, for example. It's only up in cluster 0, and it was one of the tops. So basically, we can maybe use this to annotate them. So I could quickly draw the heat map too. Heat map may actually take some time to render, so we may actually wait a little bit, but eventually it's going to visualize. But you have something like this as a result, if you wait. So basically, if the color is yellow, meaning is that the expression of this gene in this cluster is quite high, which is indicating that these genes in this cluster are unique to these clusters, because as you can see, along the row, as compared to all other clusters, none of them are actually up, right? So this is quite an isolated cluster, right? But these genes are unique to these cluster 0, but it's actually sharing some other genes that are significantly up in cluster 0, as opposed to cluster 9 as well. So if you go to resolution 0.6, what you'll see is that cluster 0 and cluster 9 are so close to each other, meaning is that they are sharing some kind of markers, but there are markers to separate them as well. So this will be some kind of like a typical heat map where some of these genes will be specific to some clusters, but some of these genes might be shared across clusters. And this is actually quite true because some cell types and subcell types are actually sharing markers, even though they have specific ones and signals to them as well. So the other thing, for example, as you can see over here, is that this is probably the T cells that we are looking for. So there are multiple of those where there are some genes that are shared across 1, 2, 3, 1, 2, 4, and 6. But some of these genes are higher and lower based on where you're looking for. And that is what we are trying to find. So basically, this is an iterative method. It's not like you can just pick it up by looking at the heat map alone. You have to look at the you have to look at the table. You have to look at top 10. Maybe you missed some gene by picking up top 10. Maybe you have to increase that number to 20 to pick it up. It makes sense. It might be the case that the gene that you're looking for in order to identify that cell type may not have as high log-fold change as these top 10 have. So it's based on how you play with the data and try to understand what makes each cluster different. And again, this may not be the actual number of clusters that you may have. You may have to need to keep, dividing it, looking at the gene expression that you have, and keep trying to figure out different subtypes of cells that you may have. And there are other ways to look at the identities of clusters too. So, for example, I may need to pick up some markers over here. I'm going to get CD8A somewhere, even though it's not here. Okay, CD8A, say C. So there are some CD8s genes that are up in some cells. So I'm going to search for that. I'm going to search for granulysin. CCR7 is usually found in naive T cells in both CD4s and CD8s. And PPBP will be usually found in the platelets available in the PBMC. So you can draw a violin plot to see the gene expression of each cell in each group to have a better view of what each cluster is. Oh, this is the future plot. Oh, this is the violin plot, yeah. This will be the one. I'm going to show you a future plot as well. So basically, what's happening over here is that there are 3 clusters that could be potentially CD8 T cells that we are looking for. It's 4, 6, and 8. And we have 2 clusters that, some clusters significantly have granulysin, but one is actually higher than that. So basically, this could be the NK cells, and these could be the T cells that we are looking for. Then we have some CCR7s. So what I can see is that 8 is sharing both CD8A and CCR7, meaning is that the cluster 8 could be the naive CD8 T cells that we are looking for. And again, the cluster 10 has the highest expression of PPBP, which is quite clearly indicating that these are the platelets. So you can do the same thing in future plots. You can basically do the same visualization from a different perspective by using feature plots. You're basically doing the same thing. You're using the same features. You're determining that you're going to use the Tisney reduction, and just click on it, and it's going to give you multiple feature plots, Tisneys, that tells you which cluster actually. I can't zoom in. Okay. For some reason this is not working. Yes, there you go. So cluster, is this 6 or 8? This is 6, this is 8, and this is 4. Basically, what I'm understanding is that 6, 4, and 8 are the CD8 T cells, right? So among these clusters, 6 actually have granulysin as well. This is probably the cytotoxic CD8 T cells. And again, if I take a look at the CCR7, This is CCR7 is up in both 2 and 8. There are some over here, but usually what you're trying to look at is that if where the signal is that has the most expression. Basically, you can some random number of maybe technical artifacts or some alignments to CCR7 in other groups. But what we are usually looking at is trying to find is that where it is the most expressed, right? So I'm just probably thinking that 8 is CD8 naives, 6 is CD8 cytotoxic, and so forth. So you may still need to go with 4. So again, if you are wondering what is the difference between 1 and 2, you can run additional commands as well. Basically, what you can do in this case is that 1 to 1 analysis. You can actually pick each cluster up from each resolution. In this case, you want to check if what is different between 1 and 2. And we're going to check if what is the log fold change of each gene of 1 divided as opposed to 2 from resolution 0.6. So it's not like you may be able to identify the markers just by looking at defined all marker analysis. Sometimes you may actually miss some of them because you're comparing 1 against all. Maybe some markers could be revealed by focusing on 2 and just comparing them with a simple differential expression analysis, and it's going to take much faster because you have much less number of cells. And you can view the same table here as we have done before. Now you can basically, there are no genes over here or cluster identification because you actually have one cluster as opposed to the other. The only thing that you need to search for is which of those genes are up. So basically what I'm saying is that cluster 1 actually have a bit more, what as opposed to, probably 2 was naive as we picked up. So this could be some, I don't know, some CD4 T cells where KLRB up and some S100A4 up as well. So again, the annotation is up to you. You can either annotate a cluster based on the gene that you think it's up. You can just say that it's KLRB +1 CD4s, or if you're making sure that those markers are definitely indicating some known absolute cluster type, you can just name it so. So the naming procedure is actually depending on you. So these were the names that I picked up based on all the table analysis that I did. But again, this is up to you. So here I have a simple table of all the clusters, all the significant markers that I found for each of those, and what do I believe that it's actually meaning to me. Again, this thing is absolutely up to you. So now you can use this information to rewrite the clustering labels a little bit and to see if you can turn 0 into CD14, 1 into memory CD4 for further visualization. Let's say that you are reporting some kind of an analysis to a group of people in a presentation. Of course, you're not going to use numbers like 1, 0, 2, or 3. You're going to pick up some annotation that making sure that your audience can actually understand what kind of markers can be found in each of these clusters. So you can basically do this naming with a few number of code. So with the order from 0 to 10, I have ordered all of these clusters and giving them new names. And these names are totally up to me. Again, don't mind that I've given these names. If you believe looking at some genes, you have other ideas about the identities of cell clusters, you can go ahead and do so. So what we are actually doing is that we are giving the clusters a new name by determining a set of labels, then changing the level of these vector, and then use that to give new items. So if you visualize the dim plot now, you are going to have each of these labels visualized on the testney as you're viewing it. So basically, guys, this is the most essential kind of analysis that you can do with single cell. What we have done was that we picked up genes, we filtered out some cells that we believe could be hindering the analysis. Then we started clustering. And after clustering, we tested the significant changes of those genes within each clusters as opposed to the other ones to make decisions about the identities of each cell type. Again, we talked about a lot of stuff like choosing the correct number of genes for variable features, choosing the correct number of PCs, and choosing the best resolution or the set of resolutions that we can work with in order to identify groups. So each of these steps, we broke it apart because each of them is really essential for making important decisions about the populations that you are trying to find. And one of the most important things we didn't touch base over here because it's a little bit more advanced is that once you have all these subtypes, you can actually test, let's say, CD8 effector T cells across different conditions. When you have maybe 2 samples and healthy control and a disease condition or a experimental condition, you can actually see what is actually changing across effector CD8 T cells in a healthy condition as opposed to a experimental condition. But before that, we have to do this essential analysis all the time, regardless of the fact that we only have 1 sample or multiple samples. So any questions before we finish? And I'm going to just talk about some other stuff too.
I had a question, Arthur. So this is Bruce. On the table you had up a minute ago, which was genes within clusters. Yeah, that one. I'm looking at some of those p-values. They seem so extreme. I can't even understand what they're telling me. Like the first one, 10 to the minus 209. I mean, what is that even telling me?
Yeah, it's telling you that the mean expression on all other clusters are so low as opposed to the mean expression of this gene in that particular cluster.
Okay.
So that's by the way, I mean, Bruce, you actually asked a really good question. So find all markers function has a default test, which you probably know. It's Wilcoxon test, but you can actually change it. So.
Okay.
So you can go for help find all markers.
So that would be, okay, if you're using a non-parametric like Wilcoxon, that would explain those extreme p-values because you're looking at every pair against every pair, and if you've got 30,000, it mounts up pretty quickly. So I gotcha. Okay.
Definitely. But again, I mean, sometimes, Bruce, thanks for saying this, because when you use a different test, you may actually get a different result.
Sure.
A lot of people will be satisfied with the default Wilcoxon t-test, but as your cell populations become more complex and when you have more clusters that are looking so similar to each other, you may actually need to change this test a little bit. And based on the data that you have, that might be the case. You can go ahead, do a t-test as well. But a lot of people, especially if they are not as sensitive as to Bruce with p-values and statistical tests, they may actually just say, I can go with the default one. But yes, it actually gives you that option too.
I would think that in terms of relative ranking, it's going to stay the same regardless, pretty much regardless of what test you use. But it's just the extreme level of that p-value struck me as being, if I saw that on one of my output, I'd say the thing didn't run, it blew up. But now I see why it can be that extreme. So yeah, thanks for that explanation.
Okay. Any other question?
Yes, Arthur. I have a question actually. Thank you for your helpful class. I just saw in the literature that in some studies they sort out some cells and do the single cell analysis on those sorted cells. And I think I have a project on just keratinocytes. I was wondering if I need to cluster out keratinocytes and then do the single cell, or do the single cell on punch biopsy samples.
What do you mean do the single cell?
For me, it's important to be able to cluster grouping different types of keratinocytes, and because we know that we have different.
I believe you're referring to the fact that if you have to FOX sort the keratinocytes from your sample, then do the single cell library
Yes.
prep? Okay,
Yes.
I see. Yes, I mean, in some cases, if you are really interested in only keratinocytes to achieve more sequencing depth, yes, that would be the correct way to do it. And we do that all the time, by the way. I mean, it's not like we start with all the cell types in the body. Sometimes, most of the time, when we are just interested in CD3 T cells, but we just go, sort for CD3 and prep the library, and we only have T cells, and we pick apart which of those T cell subtypes that we have in our analysis.
Okay, exactly. I want to know that it is because of the, I don't know, expenses, or even if we include all the cells, the depth is not enough to get all the clusters in that group. I don't know the reason why they sort out, they target actually cells.
Yeah,
Why
well, to get more sequencing depth, right? Because you have a more targeted population. You're gonna have RNA from just only that.
Okay.
So that is basically the reason. But again, I mean, this is a question of the quality of your sample, the objective of your study, right? So basically, if you're looking at interactions of multiple subtypes, it doesn't really matter that you have only T cells. You have to have other clusters as well. So it's basically up to your experiment. But the assumption is that now you have some group of cells here. You're just trying to find out what kind of subtypes that you have in your data.
Yeah. Thank you.
Yeah. Anything else? Anyone else? No? Then before finishing, I'm just quickly going to show you an additional analysis that you can do, which we have implemented with Alper a lot. So basically, as you can see, even though this is not a linear problem, you may still need to have some kind of a template for yourself so that you can play with it, because sometimes you have a lot of functions over here that you have to keep, pick up, copy from somewhere else, right? But what we have done with Dolphin Next is that we have come up with a pipeline that does this analysis and initial analysis with some parameters that you can choose, and you can run that, and it actually gives you some kind of results of clustering, and it actually tells you, and it actually gives you a functional, a script that you can have all the functions that you need so that you can change it later. So I'm going to just go to, Dolphin Next really quickly and going to pick that apart. I'm going to pick it. Yeah, this one. So this is a special pipeline that we wrote here in Bioinformatics Core. What's happening is that it's exactly taking those barcodes and this values. Let me just go back to that really quickly. So the pipeline, what's actually it's taking is that it takes this entire folder and it runs the analysis based on the parameters that we have used during the entire workflow. Like, what is the mean amount of UMI that you believe that each cell should have? What should be the number of variable features? What should be the threshold mitochondrial ratio, and so forth, right? And what's going to do for you when it's completed is that it's going to give you some results with plots, which give you heat maps based on all the clusters that you have found. So that you can take a look at it. So this is kind of an initial step. You can just take these workflow again and run it again with different parameters. But the most important thing is that you can actually, I'm closing this, use an R Markdown file that is over here and directly copy to your file over here and create a new R Markdown file. Let's give it main R Markdown. Delete everything is copy. So basically it follows all the steps that we have taken, right? It just used all the parameters that we have. Now you can just run them or change them. What's actually doing is that it creates some kind of reproducibility, right? Let's say that you have written some paper and you want to distribute the order of all the functions that you have used, which is really important because if you write a paper with Surat a pipeline or Surat package was being used, they are probably going to ask you which functions that you have used. So basically you can actually use this Dolphin pipeline to reproduce and distribute the functions and the orders that you have used in it with comments and which will be transformed into an HTML format. So you can run all of these in the same time or even change it. And the pipeline will actually give you an HTML version of it as well, where all the steps of the analysis with the visualizations and the plots can actually be observed. All the variable features that we picked, some number of dimensions that we chose initially, and the clustering. So I choose some different parameters here, I believe. So the clustering is somehow similar, but the shape of the t-SNE is a little bit different. But basically, it's just picking apart the same kind of subtypes that we have just found. So this was it from me today. Thanks a lot. I hope this was really useful to you to understand the essentials of single cell analysis. And we will be really glad if that you understand and try to use single cell analysis in your papers and publications as well. Because it's kind of easy if you understand the essentials of it, right? There is some kind of a workflow that you have to follow, but each step has its own logic that actually helps clustering your the data more, which better and accurately determines the subtypes that you have within your single cell library. So thanks again. Alper, do you want to add anything?
Arthur, I just wanted to give a small quick feedback. It's like, thank you very much. I want to thank you and Alpha for this wonderful bootcamp because I'm a computer science person. I'm basically deep learning, machine learning. I do with those algorithms. And I didn't have any clue about this bioinformatics, but this bootcamp has really given, I have learned a lot and thank you guys. And I'm just looking forward, to implement all these things in my upcoming works. Thank you a lot.
Sandhya, we talked about you with Alper the other day as well. So if anything we can help to get you moving forward with biology and the reason for all of this and how we are doing that, we'll be glad to help you.
So yeah,
thank you.
So we can, so we have this Dolphin Next now to help the people to create their pipelines. But if you want to develop your own pipelines, we are going to be happy to help you there too. There are lots of people now started developing their own pipelines and they're asking questions to us, like when they're creating their processes. We are helping them, and after one point they do everything themselves. Like we know some people that they never ask questions anymore to us, they do everything themselves. So please don't hesitate to send us emails, and if you have larger projects, you can also collaborate with us. We can help you to keep your metadata in our metadata system. In the future, if you go to another university, you can still use these tools. Like you don't need to log in Dolphin Next umassmed.edu, but you can install it to your own computer, or if you have a budget in the future, you can buy a server and install there, or you can install on Amazon, use everything on Amazon. There are people doing that too. So please keep in touch. We'll be happy to help. And if you have questions, you can use Slack channels or email us. Thank you very much for joining all the bootcamp. We're going to organize new ones with new topics. So hope to see you again. Bye.
Bye-bye, guys. Thanks for
Thank
coming.
you.